And thank you for the introduction, and thank you so much for asking me to talk to your KCNQ2 peer group at this fantastic symposium. So the aim of my talk today is to help you understand why we see so much clinical differences in individuals who have variants within the same gene. So I'm taking it you can hear and see my slides before I start rambling on. Okay, great. Okay. Yes. So I'm going to start by talking about two families, family A and family B. So and in these families, people who are blue have epilepsy. So family A has a 10-year-old boy who started having seizures at 3 days, and the seizures are very frequent. And despite the fact that we tried multiple different types of anti-seizure medications, his seizures continued, and he continues to have frequent seizures, including status epilepticus at the age of 10. And he is now severely impacted. He has severe intellectual disability. He's not able to walk, and he hasn't got any language. And we identified that he had a pathogenic variant in KCNQ2 as a cause for his severe developmental and epileptic encephalopathy. And I should say that this variant was only found in him and wasn't found in his parents. So this is family B, and they have two people in their family who have epilepsy. The dad, who's 45 years of age, and he started having seizures at day 3, focal seizures, and he didn't have that many of them, and quickly they came under control by day 5, and at 2 years he stopped having any anti-seizure medications, and his development was entirely normal, and he went to university, he finished a professional degree. He had a child who is now 10 years of age, who started having seizures at a similar time to his dad at about day 2, and they were focal seizures. Took a bit longer to get control of his seizures. He had a few more of them, but they did stop by 5 weeks. And at 18 months, his anti-seizure medications were weaned, and he did well until he was 6, when he started having a few more seizures, not that many, but over the next couple of years, and he's still on anti-seizure medications at 10. He's got a normal IQ, but he has had some mild learning difficulties at school. And genetic testing was able to identify that the cause of their epilepsies was also KCNQ2. So we have these two families, family A and family B, who both have epilepsy in their family caused by the gene KCNQ2, and yet they are clinically very different. So in family A, the 10-year-old boy has very, very severe epilepsy, whereas in family B, dad had mild seizures and really nothing else, and his son had relatively mild epilepsy, but it carried on and he's had some learning difficulties. So when you see this sort of clinical variability within a gene, it's called clinical expressivity. And the formal definition of clinical expressivity is the degree to which a phenotype is expressed by individuals with a particular genotype. Okay, so that's fine. But if you don't know what phenotype and genotype are, it might be a bit difficult to understand. So I'm going to talk a little bit about that. So what is phenotype? When people in genetics research or genetics in general talk about phenotype, they're talking about the observable characteristics of a person. And those observable characteristics can be physical, they can be developmental, they can be behavioral, or they might be at a cell level, so it could be physiological. And when you think about clinical expressivity within an epilepsy syndrome, or particularly in the developmental and epileptic encephalopathies, there's lots of different signs and problems that can be very variable. So you can have seizures. You can also have difficulty with intellectual function and motor ability, including movement disorders. There can be issues with behavior, such as attention deficit disorder or autism spectrum disorder, psychiatric problems, such as depression and anxiety or psychosis. And there is other non-neurological features that people with these genetic developmental and epileptic encephalopathies can get. And the variability within each of these can be quite significant. So if we just look at, say, seizures, individuals can be different with regard to when they start having seizures, how frequent the seizures are and how long they last, the type of seizures that the individual has, how it's talked about the duration, and as well as how well they respond to treatment. So there's a lot of variability between seizures in individuals with these developmental and epileptic encephalopathies, as well as other symptoms that they have. So that's the phenotype. Now, genotype refers to the genetic makeup of a cell. And so because we're made up of cells, the genetic makeup of individuals. For those of you who, it's been a while since you, I'm not quite sure because I didn't listen to, I'm sure Ingrid's talk was fantastic. She may have gone over this, but I'm going to give you a little brief summary. So hopefully you know that your body's made up of cells, and we all have about 30 trillion of them. And most of these cells have nucleuses, which is where we find the genetic material. And this genetic material is found in very tightly woven coils of genetic material called chromosomes. We have 23 pairs of those. And as I said, they're made up of very tightly wound up DNA. And DNA is made up of base pairs, and you've got about 6 billion base pairs. And each of these letters, A, C, G, T, represent a specific base. And these are in order, the letters are in order, and that makes a code that is really important in terms of making us who we are. So a series of these codes, or a length of these codes that code for a protein, is what we call a gene. And you've got about 20,000 genes in our DNA. So that's what's going on in terms of your genotype. So clinical expressivity, remember, is the degree to which a phenotype is expressed by individuals having a particular genotype. So why is it? Why is there so much difference, though? Now, humans, genetics is complicated, and it's very complicated in humans. So I'm going to try and make it simple for you by talking about circles for a little while. So this is a circle, and it has a gene Y, which is normal. And the phenotype, or the physical characteristic of this circle, is gray. Okay. So if you have an abnormality in gene Y, it might result in a different phenotype of this circle. So before I go into that in a bit more, I want to talk about this word abnormality first, okay? So what do we mean when we say an abnormality? What we mean is a change in the gene, okay? And something that is different from normal. And we call these changes variants, okay? And if it's a variant which causes a disease, we call it a pathogenic variant. So when we use the term pathogenic variant, that's what we're meaning, a change in the gene that causes a disease. So back to our circles. So our genotype Y here is normal, and you have a gray circle. But if there's an abnormality in that gene, so a variant, it can cause a different phenotype. So here we've got 3 new phenotypes, okay? So purple, green, and blue, which are caused by variants in that gene Y. Variant A, variant B, and variant C. So what does that mean? What's going on? Okay, let's go back to our genes again. So here we've got a gene, and it's got a series of base pairs, and it looks here like there's not that many base pairs. That's in this figure, that's a bit misleading. In reality, each gene has thousands of base pairs, and in KCNQ2, for example, there's over 60,000 base pairs. So that means there's an awful lot of different places that mistakes can be made. So what happens when a mistake is made? So if you get an abnormality in the code, well, a lot of, so first of all, what do those genes do? So those genes, okay, that code codes for a protein, and you have factories in your cells that link onto the gene, read that code, and make these proteins. And here is a picture of showing a structure of a protein. And in KCNQ2, the protein that's made is in a channel, which is on the cell membrane. So this isn't a potassium, sorry, it's a potassium channel. This isn't a potassium channel. This is actually a picture of a sodium channel. But a similar thing happens. The protein finds its way to the cell membrane. So here you have a cell, you've got the inside of the cell and the outside of the cell, and this is the cell channel. And in this particular protein is the alpha part of that cell channel. And so it sits in here, and this sodium channel is responsible for making sure there's the right amount of sodium inside the cell. Okay, so this is the normal situation. So what happens if you end up with a problem in the code? Well, let's say here, there's a mistake, a change happens. Well, a lot of different things can happen. One thing that can happen is it can mean that the cell machinery can't hook onto this gene at all and just can't make protein at all. So you have no protein, it can't go into the cell membrane, and you end up with a channel that's really abnormal and doesn't work at all. So that's bad. But it isn't always, the changes in the gene don't always mean that the cell isn't able to make a protein. So let's take another example. Say this little red X here, you get a variation here, and maybe that only changes this protein a little bit. So it still may, but it's not got quite the right structure. So it's a little bit of an unusual shape. It still gets its way into the cell membrane, but the channel isn't quite right, and it doesn't work right all the time. And that might mean that the channel has a loss of function. So it's not as good as doing what it's supposed to do with the sodium channel. You might have another variant, let's just say here, let's go for the green one, and it might impact in a different place in the protein. And again, this protein makes its way out to the sodium channel, but isn't quite right. And again, the structure, therefore, the function of this channel, it doesn't do what it's supposed to do quite correctly, but it might mean that there's a gain of function. Now, you might think that gain often sounds like a good thing, but these channels are, made just perfectly. And so both a gain of function and a loss of function isn't good. What you want is just the right amount of function. So back to our little circles. So you can arrange, and we've got our three phenotypes, purple, green, and blue, caused by variant A, B, and C. And you can imagine that these variants are all at different places in gene Y, and they impact in a different functional effect, and they result in this different phenotype. So different variants in different places within the gene can cause different functional effects for the protein and a different phenotype. So that's one reason why you get different clinical features in somebody who has an abnormality in the same gene. And we'll give you an example of this in the gene SCN1A. And so here, each of these different colored stick people represent a different epilepsy or a different phenotype associated with pathogenic variants in this gene. And so pathogenic variants in SCN1A can cause a lot of different disorders, can cause a very mild type of epilepsy called genetic epilepsy with febrile seizures plus, where people might just have seizures with fever when they're children and nothing else. It can cause more severe types of epilepsy, myoclonic atonic epilepsy, or very severe epilepsies such as Dravet syndrome, early infantile encephalopathy, and epilepsy of infancy with migrating focal seizures. And in some individuals with pathogenic variants in SCN1A, they don't have epilepsy at all, but they have autism spectrum disorder, or they could just have hemiplegic migraine. So you can get a lot of different disorders when you have when you have variants in this one gene. I'm going to talk about two of these disorders in more detail: Dravet syndrome and early infantile encephalopathy. So Dravet syndrome, these are both not good epilepsy syndromes to have. But early infantile encephalopathy is much more severe. So Dravet syndrome presents with seizure onset around about 6 months. And in second year of life, children start having developmental delay, and they end up with mild to moderate intellectual disability. Whereas in early infantile encephalopathy, they have seizures at a much younger age. They start having developmental concerns by the time they're 3 months, and they have a much more severe outcome with profound disability. So these children aren't able to walk, they don't talk, and they need G-tube to get nutrition. And they also have this very significant persistent movement disorder, which impacts negatively on their quality of life. So same gene, but two very different disorders. And if we look at, so they also are caused by different variants within this gene. And early infantile encephalopathy is mostly caused by a single variant. And our colleagues in Melbourne, Steve Petrie, who are going to hear talk about later in the symposium, and his lab were able to have a look at what those variants did. And they put the variant into a cell, and they showed that the variant increased the function of the sodium channel. And that was very different to previous research, which had shown that variants that cause Dravet syndrome cause a loss of function. So here's an example where you have the same gene, but you have two different variants. One of the variants causes a loss of function, or one type of variants cause loss of function, and you end up with Dravet syndrome. And another types of variants cause gain of function, and you end up with a much more severe type of epilepsy. So that's, as I said before, that's one reason why you get differences between individuals, why you've got purple circle, green circle, and blue circle. So if that's the only explanation, then you would expect that everybody who has this variant here should be the same, okay? Unfortunately, that's not the case. And what you end up with is, even within these diseases, you get individuals with very mild phenotypes and individuals with more severe phenotypes sometimes. And so I've represented this with different shades of colors of these circles. But if we look at this in a human example, this is a study on a gene AGL13, and that causes a severe developmental and epileptic encephalopathy. And in this study, which was published last year, they looked at 38 individuals who all had exactly the same variant, so the same change in that gene. And when they published this, and so even though these individuals had the same gene and they had the same variant in the gene, they still had quite a different clinical outcome. So 10% of the individuals, so what was the same about them was they all had infantile spasms, so that's a particular type of epilepsy, but 10% of the individuals ended up with mild ID and were able to walk and interact, whereas almost half of them had a very profound outcome with profound intellectual disability and were unable to walk and talk. So this is an example of the same gene, same variant, and yet really different clinical features. So, why is that? So there's a few explanations. There's quite a few reasons for this, and I'm going to focus on 3 of them today. And the first one I'm going to talk about is genetic load. And even within genetic load, there are different reasons why individuals may have different levels of genetic load. But the example I'm going to give you is mosaicism. So what's mosaicism? All right, so you'll remember that you're all formed by a sperm from your dad and an egg from your mom, and that's where your genetic material comes from, and that gets together and forms a cell, and that cell replicates. And remember, you've got 30 trillion of those cells in your adult body, so it replicates a lot of times. And sometimes what can happen is a mistake is made. In fact, it's miraculous that more mistakes aren't made, given how much copying goes on. But so in this example, what's happened is at a 4-cell stage, we've got a mistake has been made, and the normal phenotype of these cells is purple in this example, but there's been a change, a pathogenic variant or a mutation, and it's resulted in orange cells. So the genetic there's a change here in the genes than from the purple cells. But these cells have to keep replicating over and over and over again to become a human. And so the purple cells keep replicating, and their DNA is the same in all of their cells, and so do the orange cells. And so what you end up with is an individual who's got 2 different groups of cells, purple cells and orange cells. So they've got a variant in the gene that's making them mosaic. So they're mosaic for this genetic abnormality. So that's what mosaicism is. So what's interesting, if we look at these 2 families, is that in individuals who are mosaic for a genetic abnormality can sometimes have a milder epilepsy phenotype. So if you have less of a genetic load, you're less severely affected. So this is an example of 2 families, both who have mutations in pathogenic variants in SCN1A in their children. So all of the children, so the purple children here all have Dravet syndrome, and they have a pathogenic mutation in SCN1A, which is causing their Dravet syndrome in every single cell in their body. So they are not mosaic, okay? But using quite sophisticated genetic testing, they were able to show that in one of their, that these children actually inherited that variant from one of their parents. But their parent was mosaic for that variant. So in this parent, there was only 28% of the cells that had the abnormality in SCN1A, and in this parent, there was only 17% of the cells that had the abnormality. So these parents were mosaic for this SCN1A variant. And they did have epilepsy, but they had a very much milder epilepsy. So they had febrile seizures, so just seizures with fever between the age of 6 months and 6 years. So this is an example of where and less of a genetic load. So if you're mosaic, you've got less of the genetic abnormality in your body, you have a milder phenotype. So that's one reason why you can, so genetic load is one reason why you can see variability in somebody who has a variant, exactly the same variant in a gene. But that's relatively uncommon. What's much more important in clinical expressivity is the genetic background. Now, what is the genetic background? So remember, it's all very, you can have a pathogenic variant in a single gene, say KCNQ2, but remember you've got 20,000 genes in your body and they don't all act in isolation. So all these genes interact with each other, and it's these genes that make your genetic background. And the other thing to remember is that, you and I are very different. Every single individual has a unique genetic code. So my genetic code is different from everybody else in the world. So that means I have changes or differences in my genetic code from everybody else in the world. Now, some of these changes are important just to make me me. So they're very normal, but they make me different from you. And others of these changes can be disease causing. But it's these changes in the genetic code in the 20,000 other genes, which is what makes up your genetic background. Now, when we look at these changes that can lead to disease, they tend to get grouped into two main categories. So there's rare variants and common variants. And so what do we mean by rare and common? So we're really fortunate now that we have over 100,000 healthy individuals or individuals who have been had their DNA sequenced, and it's put in these big, massive databases. So when we can look to see how common a change is in the population. And so some of these changes are, that an individual might have can be very, very rare, and some of them can be quite common. And we can tell that by looking at these big population databases. So if we look at the rare variants, so rare variants are very rare. So that means you don't see them in the big population databases. They're not seen in the healthy population. And it's these rare variants that can cause disease. So like such as in KCNQ2 and in SCN1A, as I was speaking about earlier. And these rare variants can have a major effect on the protein, and that's why they cause disease, okay? Common variants are frequently found in the database, and what that means, and they're found in healthy people. So what that means is that although these changes may have a small effect in the protein, a very small effect, on their own, they don't cause diseases. It is possible that if you have multiple variants, which cause a small effect in a particular area, that can lead to disease. So if you have multiple common variants that might impact on genes that are important in epilepsy, the addition of all of them together can increase your risk of developing epilepsy, or can sometimes increase what's responsible for the genetic background that is important in the variability that you find in somebody who has a rare variant that's causing their epilepsy. So let's look at that in a little bit more detail. So rare variants can either be disease causing variants, that like say cause a KCNQ2 encephalopathy, or they can be what we call modifier genes. I'm going to talk about that in a bit more detail. And common variants contribute to what we call a polygenic risk that somebody has of developing a disease or impacting on the variability of the clinical phenotype they have within a disease. So unfortunately, there aren't a lot well, there aren't any good examples of modifier genes in an epilepsy situation, and so I'm going to, but there is some in mice. So I'm going to tell you about this report that was done in 2020. And what this group did was they put pathogenic variants in a gene called SCN8A into 2 different strains of mice. So SCN8A is a sodium channel gene, which also causes epileptic encephalopathy. Well, it causes epilepsy, and just like KCNQ2, some individuals can have very mild epilepsy, and some individuals can have very severe epilepsies. And what these researchers did was they took a particular SCN8A variant, which causes epilepsy in humans, and they put it into these 2 different strains of mice. Now, what is a strain of mice? So researchers who work on animals, they generate these mice strains, and what that means is that they are very pure genetically. So every mouse within a particular strain is similar in their genetic background to all the other mice in that strain, and different from mice in different strains. So basically, different strains of mice have a similar genetic background, and because their DNA sequences is very similar. So they're the perfect situation, or the perfect animal to look at, a way to look for modifier genes. So the researchers put a variant in SCN8A in both of these different strains of mice. And what they found was that in one of the strains, they had a very severe phenotype. So they had early onset of seizures and died shortly after that. But in a different strain of mice, even though they put exactly the same mutation, the change into that mouse had seizures much later, much less frequent mice, and they lived much longer. So the researchers were able to look at the rest of the genetic background to see, well, what's different in the background of this mouse that makes it so much better than this one, even though they both have this same abnormality. And what they found was, what they found was a variant in a gene called GABRA2. And they found the variant in the mouse with the better outcome. And what they found was that there was a variant in this gene which impacted on the protein, so that the protein was expressed by much lower, about 75%. And so, but that in this mouse, whereas this mouse that had the normal GABRA2 had a normal amount of that protein. And so basically what's happening here is this variant in this gene, GABRA2, is modifying the severe phenotype for SCN8A. So it's protecting this mouse from having this severe phenotype. So this is an example of a modifier gene. GABRA2 is modifying the clinical phenotype of SCN8A. So I'm just going to show you that in, So I'm going to show you that in this figure. So this figure, let me take you through it. This is about variants, okay? So the x-axis here is the frequency of the variant in the healthy population. So if a variant is at this end, it's very rare, and if it's at this end, it's very common so these are the common variants, and these are the rare variants, all right? And this y-axis is meant to demonstrate the effect that a variant in these variants are having on the protein. So the likelihood they are to be causing a functional impact and change the phenotype. So, and the size of the circles also reflecting that. So we have this gene X here, okay, which is a rare variant because it's at close to this side, and it's also large and high up. So it is impacting on the protein and the function. And so let's just say that so I'm going to talk about 3 different individuals. So individual A has got a variant in this gene X, this rare gene, which is causing a disease, phenotype A. Now, this variant in this gene is enough to cause this disease. Let's say it's an epileptic encephalopathy all by itself. And this person is relatively lucky from a genetic background perspective, in that they don't have any other variants which are impacting on their phenotype. And so they have this phenotype A. In the modifier gene situation, like we just discussed with the mice, this is what you would see in individual 2. So individual 2 here has the same abnormality in gene X, but in their genetic background, they also have a modifier gene, which is impacting on the phenotype. So their phenotype is different from individual 1. Now, the final situation I'm going to show you here is this third individual. Okay, so the third individual again has an abnormality in gene X, but they don't have any abnormalities in any modifier genes, but they have got lots of common variants that are impacting on their phenotype. So they develop this third phenotype, phenotype C. So you have these three individuals, and what their phenotype ends up looking like depends on what genetic background they have. Individual 1 has just got the main mutation, individual 2's got a modifier gene, and individual 3 has got some of these common variants. So if we're now talking about just these common variants and how they impact on disease, there aren't any yet examples of that in terms of an epilepsy perspective. So I'm going to take you through a cancer example. So this is relating to breast and ovarian cancer, and I'm going to take you through this diagram in quite a bit of detail. So across the x-axis, you have what they call the polygenic score, or the polygenic risk score. And what that basically means is the number of common variants that the individual has that in themselves don't cause a major effect, but when you have lots of them together, they may have an impact on the disease. And then this y-axis is the probability that somebody will develop cancer by the age of 70. So, and then there's two lines, a blue line and a black line. So the blue line is the situation where you have an individual who has a mutation in a gene of major effect that causes cancer. So they have got a rare variant, which on its own can cause cancer. And the people in the black line don't have any abnormalities in the genes that cause cancer all by themselves. Okay, so if we look at the black line, so the individuals who don't have any genes that on their own can cause cancer, and we look at an individual here who has no common variants that increase the risk of cancer, you can see this individual's chances of having cancer by the time they're 75 is really low. But if they have the more and more common variants that the individual has that impact on cancer, the higher their risk goes. So that if you have all of the risk factor common variants, the risk is about 30% of having cancer. However, if we take somebody who's inherited a gene of major effect that can cause cancer all by itself, and we look at the impact of having common variants as well, it's quite remarkable. So here's an individual who has inherited a gene who can cause cancer all by itself, but has no common variants that cause cancer. And so the risk of cancer is relatively low. Well, I mean, it's much higher than somebody who doesn't have that major effect, but it's still down in the sort of 12% range. But for an individual who has that variants in that gene, as well as lots of common variants, their risk factor goes all the way up to 80%. So hopefully you can understand from this figure how having multiple common variants can impact on a disease. So that's your genetic background. So your genetic background can impact on what you see in a clinical phenotype in somebody who inherits exactly the same variant in a gene. So if that was the case, so if that was the only two explanations for why we get differences in variability in an individual who has exactly the same variant in a gene, then you would expect that if you have exactly the same genetic background, that you should have exactly the same clinical features. And so where do we have individuals who have exactly the same genetic background? Well, that's twins, because twins, egg and the sperm get together, make a cell, and then that cell splits up in a simplistic form and basically just replicates. So you get two individuals with exactly the same genetic background. So they have the same DNA sequence. So is it the case that identical twins always have exactly the same clinical features? Well, sometimes, but not always. And so this is an example I'm going to talk to you about in a gene called PCDH19, which also causes an epileptic encephalopathy. And these two twins who had this mutation in this gene both had seizure onset at 10 months. It was amazing. They had seizures almost within hours of each other. And they had exactly the same types of seizures. So that's very understandable. You think, well, you know, they've got the same genetic background, they've got the same mutation. Why wouldn't they be exactly the same? But that didn't carry on. So what happened was they actually ended up quite different. So this is a graph showing you the number of seizures that these individuals had. So in this particular type of epilepsy, PCH19, children have clusters of seizures. And what that means is that they have lots of seizures all together. So, multiple seizures every day for a few days, and then they'll go for months without seizures. So they have clusters and then long periods with nothing. Okay. So this graph along this is for both of the twins, twin 1 and twin 2, and along the x-axis is how old they were, and the y is how many clusters of seizures they had per year. And you can see at 6, we were able to identify a drug which stopped their seizures, which was fantastic, and they both didn't have any seizures after the age of 6. However, before then, despite the fact that we were treating these girls exactly the same, they were quite different. So twin 1, in her 4th year of life, had almost 12 clusters of seizures. So almost every month she was having significant periods of seizures. Whereas twin 2, the most clusters of seizures she had in a year was only 4. And when she did have her clusters, she didn't have as many seizures per day, and she didn't have as many days of seizures. So these girls are now 13 years of age, and they are very different. So twin 1, the girl who had lots of seizures, has severe intellectual disability and autism spectrum disorder. She can't speak, and she has a lot of difficulty interacting with people. Whereas her sister, who had less seizures, has mild intellectual disability, is able to read, and has lots of friends. So even though they had exactly the same genetic background and exactly the same variant in the gene, they ended up with very different clinical features. So why is that? So that is due to epigenetics. Now, epigenetics is quite complicated, and it's to do with the expression of genes rather than the sequence of genes. So let's go back to our little gene slide, and remember that your DNA material is wrapped up very tightly in these 23 different chromosomes, which is great, okay? But unfortunately, when the DNA material is wrapped up all tightly like that, your cell factories that take the code and make proteins can't latch on and can't read the DNA code. They can only read the DNA code and make proteins from the DNA when it's unraveled. So the gene is only expressed when it's open up, and the factories in the cells can go on and hook onto it and make proteins. When it's all wrapped up tightly, the gene isn't expressed. So there are various different epigenetic processes that impact on whether a gene is able to be read. So whether a gene is turned on and it can be expressed and make a protein, or whether it's turned off and it cannot be read. Some examples of that are histone modification and RNA interference and DNA methylation. And I'm just going to focus on DNA methylation. So in DNA methylation, so these little methyl groups attach to the DNA, and what happens is that the more methylation that's attached to the gene, the harder it is for your cell factories to read the code. So if the DNA is very highly methylated, then the gene is turned off and you don't get gene expression. Now, this is a really good thing, okay? When you think about it, remember that, we're 30 trillion cells, and every single one of those cells has got exactly the same code. And yet we're not just a blob of 30 trillion cells that all do exactly the same thing. We are amazing. We have got all sorts of different organs, muscles, brains, and they all have to be formed. And each of those is busy doing their own thing all the time. And that's all because they have different the genes in each of those cells are expressed differently. So you have the same DNA in each cell, but the code within that cell has to make different cells and different organs. And the way it does that is the genes are turned on or turned off. And so that's all to do with gene expression. And early on in development, that's really important in terms of forming tissues and making us, into humans. But even now, as you're sitting, listening, talking to me, genes in your body are being turned on and turned off based on what the cells in your body need to be doing right now. So gene expression is really important. That's a really important, but it's not to do with the sequence of your DNA. It's all to do with whether the DNA is being, how it's being expressed, how those genes are being expressed. So what we know is that epigenetic differences, okay, can be found in individuals, which is what you would expect, okay? And you can change the epigenetic profile, or the epigenetic profile is changed by environmental factors. So different drugs, whether they're medical drugs or recreational drugs, trauma, the things that you eat, things that happen to you in the womb, all of these things can have an impact on your epigenetic mechanisms. And so, and can change your epigenetic profile. So if we just look at twins, for example, so identical twins, remember, same genetic makeup. When they come out of the womb, if you look at their epigenetic profile, they're pretty similar, okay, which is what you'd expect. They've got the same genetic makeup and they've been in the same environment. So they come out and their epigenetic profile looks pretty the same. But as they get older, their epigenetic profile changes. And so over time, their epigenetic profile becomes different. And what we also know is that the less time those twins spend together, so the more their environmental impact is different, the bigger the epigenetic changes between the twins will be. So the implication of all of this is that your environment impacts on your epigenetic processes, and we know that can be short-term, temporary, but it can also have chronic, long-term effects. And so that can impact on how your genes are expressed. And so that is another reason why you can have different different clinical features in individuals with variants in exactly the same genes. So again, there aren't a lot of examples of this in humans, but there is an epilepsy example in a rat this time. So in this study, what they did was they had a model of inducing epilepsy using trauma. So what they do is they give the rats head injuries, and then the rats go on to develop epilepsy. And in this research project, they looked at the epigenetic profile of these in the brains of these rats before they gave them the head injury, and then 3 months later when they were developing when they had seizures. And what they were able to show is, as was expected, was the epigenetic profile was different. These mice, the genes were now being expressed differently, and that was related to how they were having epilepsy and their epilepsy phenotype. So epigenetics is important in terms of clinical expressivity. It's not involved in sequence of DNA, it's just what's important in how the genes are expressed. So now let's go back to our family A and family B. And hopefully, and remember, you've got that in family A, there's this individual with a very severe epilepsy, whereas in family B, the dad had very mild epilepsy and his son had ongoing epilepsy, but with just mild learning difficulties. And hopefully, from what I've talked to you about over the last 30 minutes or so, you'll now have some insight into why, despite the fact that all of these people have epilepsy caused by the same gene, KCNQ2, their epilepsy phenotype is very variable. So in summary, clinical expressivity or differences in clinical features for people who have disease or epilepsy caused by the same gene can be caused by many features. It can be due to just the fact that the variants within the genes are different, and so they are causing different functional changes, and so have a different phenotype because of that. So it can be where in the gene the abnormality is. It can be due to the amount of genetic load, and I've talked about mosaicism particularly. Really, really important is what's going on in the other 20,000 genes. So the genetic background, and it's important. So there can be rare variants, which are modifier genes, which modify in the background, which modify the clinical features, or there can be lots of common variants that people inherit, which can modify the clinical features. And finally, it's not just all about the sequence of the genes, it's also about how the genes are expressed. So epigenetic phenomena and mechanisms can impact on the variability that we see in individuals with changes in the same gene. So I'm hoping that you will have found this useful. These concepts that I've tried to explain are really, really complicated, even for me. And I've tried to make them simple to understand. But, you know, at the end of the day, researchers, we're only at the very top of this big genetic iceberg understanding, and a lot of fantastic and amazing research is being done by scientists, many of them who are talking to you in the symposium today, to try to understand it in much greater detail. So I hope this has been helpful, and thank you for listening.
Oh, Lynette, it's been so helpful and fascinating. And I want to point out for our families and for our scientists and researchers and others who are joining us on this call, that if you're thinking, wow, I want to see that twice or maybe 3 times to just take in all those different ways that this plays out, we're going to be posting Professor Sadlier's talk and the other talks by all our other presenters on our website. So you'll have the opportunity to go over them again at your leisure. Just a couple of thoughts as I was listening to this, and it was fascinating and extremely clear. It strikes me that your presentation just says, get everybody involved in the natural history study. I mean, that just seems the logical extension, because it's by getting those phenotypes, by getting that kind of laid out, isn't it, that we'll better understand that. Would you agree?
Absolutely. I mean, you know, it's we really need to understand the different clinical variability in as much detail as we can. So the more people that are in the natural history studies, the more we can understand that and see how much variability there is. And ideally, the clinical history studies will also allow us to have particular aspects of the clinical features, which the clinical sort of progress that will help us identify more severe phenotypes and milder phenotypes, which of course is really going to be important for precision therapy. So it is really important to have lots of people involved in the natural history. And it's also really important for the researchers to look at this genetic, and for us to try to understand genetic background and understand epigenetics, and understand what the variants in each of these genes do, so that we can make sure that these precision therapies are being used in the right individuals and are going to be as effective as possible in the right individuals.
Exactly. And that is absolutely the goal. Let me ask one question right off our group chat, and I have a couple more. Here's one from Kara. Hypothetically, if our children received whole genome sequencing cross-analysis, would there be a means to determine if there are common proteins that can predict KCNQ2 mutation phenotypes?
Absolutely. But the problem is that we don't know how yet. So it's in there. It's in that whole genome sequencing, but we don't know which of those. We don't know what the modifier genes might be yet. We know some of the polygenic risk genes, so we know some of the common variants in terms of big epilepsy populations, but not specifically for KCNQ2. But the information is all in that sequencing that your child's already had. It's just the science. We're not up. We don't know enough yet, but that's what all these researchers are doing.
Can I follow up on that? I'm sorry, I didn't mean to interrupt you, but I just want to make sure I understood you, because I wanted to ask you, are there any modifiers yet? You mentioned that earlier with Theskiana 8A, I think. Are there any modifiers for KCNQ2? Are there any areas to look at for research?
No, so that's one of the really hot areas of research. That Ingrid, who you've already heard, is really keen on identifying modifier genes, because it might be that there could be modifier in that. So remember, that was just a mice model. That wasn't in humans. We don't know much about that GABRA2 and how it modifies human SCN1A. It was just in the mice, but it's just an example of how it could work. We know there must be modifier genes. We know there has to be common variants. We just have to find them. So there absolutely could be modifier genes which impact on the phenotype when people have an individual specific variant in KCNQ2. Or absolutely.
I think one of the questions that comes up in our parent group a lot is that parents, understandably, us too, you get very, you think a lot about how a child is going to turn out based on their variant. And listening to this conversation, it really seems to me that maybe we need to pull back a little bit from that, because it's going to be hard to determine just from that variant. There are a lot of moving parts here, listening to this presentation.
Yeah, so I think tomorrow in your symposium, Al George is giving a lot of information about the different variants and how much you can tell from just looking at the variant. So of course, there are some things you can tell in some particular variant. So take the SCN1A example I gave you. In the early infantile epileptic encephalopathy, that specific variant, all of the children with that variant have a very severe phenotype, very different from Dravet. So sometimes the variant, the specific variant tells you a lot. But then in other situations like that ALG 13, all those kids had the same variant. And yet, 10% of them are pretty good and 50% of them are really bad. So there's something else going on as well. So the variant itself, Al will be able to answer all of those questions, I'm sure. But it does open up lots of questions. And, although we know so much more than we used to, there's still so much for us to understand.
Oh, well, we're grateful that you're working on it. That's for sure. A couple more questions quickly before I let you go. In terms of expressivity, and you were talking about that fascinating twin study and, the ways in which people could be influenced. You mentioned drugs. Of course, I think drugs, I think medicine. I think what we've seen in the case of our own daughter, with things that have worked, not worked, and actually changed. In certain cases, we would say how she was able to learn. Do you think medicines are going to play an important role in terms of getting the right medicines for the right kids, even if it's just a seizure med, but well beyond that, as we look towards therapeutics, treatments, et cetera?
So I guess medicines work directly on the cells to do things, so anti-seizure medicines that are sodium channels block sodium channels. So they have a direct mechanistic way they impact on seizures, and which may then potentially impact on behavior and cognition. In the epigenetic, which is quite different from epigenetics. So when I'm talking about the medicines impacting in epigenetics, so if you think about a baby in the womb, sometimes drugs that the mom is taking can change the epigenetic profile, which then changes expression. So some of genetic heterogeneity, I'm sure you've heard that before, it might be due to what the genes are doing to epigenetics in the baby within the womb. So there's two different aspects of that. So I think in terms of anti-seizure medicines, I think they tend to work more directly in the channels as opposed to changing the genes that make those channels function.
All right. Let me ask you another. Yes, it does. Let me ask another question. Someone is asking, is it correct to assume that inherited cases have 100% genetic load and that de novo cases all would have something less since they're caused by mutations after cells start replicating? You're smiling. Or are the mutations occurring early in cell division?
Yeah, that's very, whoever gave that question is very smart, obviously. So de novo mutations tend to happen really early on, okay? So, but they can happen, remember when you're looking at, when you're doing genetic testing, you're generally doing testing in blood, okay? And so that's a tissue. So what you're actually measuring is the amount of variation in the blood tissue. That doesn't tell you necessarily what's happening in the brain, what's happening in the kidneys, what's happening in the ovaries, say, okay? But we tend to assume that when it looks like, that all of the cells in the blood have the variant, that the person, therefore, the genetic load is through all of their cells. And if their parents don't have it, then it's de novo. Now, in the de novo situation, usually the change has been made really early on when those cells come together. So I guess, there could be the odd cell in there that doesn't have the variant, but in general, you've got a full genetic load of that variant. And the parents have none. But what I was going to say was that in that study that I mentioned, that was a study that Heather Method did, and Ingrid who was involved in Ingrid and New Zealand, Ingrid Sheffer in Australia and New Zealand patients. And what we did was look specifically for individuals who were thought to be de novo on clinical testing. and did really high sequencing to see whether their parents had any. And so what we identified was that some of their parents actually had small numbers of the change in their cells. So
I see.
Thank
you for
if a child
clarifying
is
that.
felt to be de novo, generally that means there's nothing in the parents. But about what we found was about 10% of or 8% of parents who were felt to have nothing in their that they didn't have a variant. When you look really closely, about 8% have got a small amount of it. So it's the parents that are mosaic. I hope I didn't get everybody.
All right. Thank you for clarifying that. Well, as I said, that was a fascinating presentation and a lot for parents to have a closer look at. But Lynette, thank you so much for joining us with this presentation. And we look forward to hearing what else comes up as you continue your research.
Thank you, Sarah. And I'm sure it's going to be fantastic talks yet to come. So